Integration of data types for inferring species biogeographic histories, genetic, fossil, and occurrence. Hello, I'm Adam Smith, and along with my co-authors Alyssa Brown, Andrea Dawson, Alan Strand, John Robinson, and Sean Hoban, I welcome you to our talk. This is but one of several talks in our organized oral session on integrating different data types, so please check out the other talks. Also, we're having a live Q&A session for the entire organized oral session on Monday, August 3rd, 3 to 3:30, so I encourage you to attend. Also, please feel free to pause the slides at any point if you want to examine something in more detail. Species have been moving across the face of the earth ever since there were species. The study of these movements and the processes that generated them is the subject of historical biogeography. More specifically, we would often like to know where species found refuge during past glaciations. When did they begin to emerge from those refugia? What paths did they take, and what mechanisms drove the changes in distribution? To that end, biogeographers have primarily used three different types of evidence and respective ways of analysis to infer the history of Earth's species. The earliest type of evidence used was fossils, and I'm going to focus specifically on pollen because later we'll be talking about trees. Fossils provide us evidence of historical presence. That's the strength of this type of data. Unfortunately, fossils and pollen can usually only be resolved to the level of a genus. Likewise, it only gives us relative abundance, not absolute abundance, and because pollen can distribute itself much more widely than adult trees can, in some cases you'll have false positives, but in other cases you might have lack of deposition even though the species was present in the surrounding area. Biogeographers also employ DNA to understand biogeographic history. Genetics can tell us about how different populations are related. It can give us population size. Unfortunately, it also suffers from an identifiability problem. For example, areas with high genetic diversity today may have arisen because they were the refugia from which species emerged after the last glaciation. On the other hand, they can also arise when clades from different refugia merge together. Finally, biogeographers also use modern-day occurrences to train species distribution models, or niche models, to understand the location of suitable habitat both in the present and the past. While powerful, these kind of tools rely on many different assumptions. So as you can see, each data type has different strengths and different weaknesses. Having recognized the various strengths and weaknesses of these methods, ecologists often combine them to infer things that they couldn't if they were to rely on one method alone. I want to give you an example of that, and I'm going to be a little bit critical so I don't feel fully comfortable critiquing somebody else's work. So this is actually based on something that I helped out with. We are interested in examining the biogeographic history of Delphinium exultatum, a critically endangered plant. And genetically, we know that it's divided into a highland population largely relegated to the Appalachian Mountains and to a lowland population. My geneticist colleagues asked me to construct niche models for this species because they knew from the genetics that these two clades had split about 6,000 generations ago. However, some people say the generation time is equal to 1 year, whereas other people say it's equal to 3 years. To resolve the issue, I constructed niche models for the species. You can see projections of those models on the right for the present day. And what we found was that past 10,000 years ago, suitable habitat for the highland population basically didn't exist, suggesting that the generation time of the species is somewhere on the order of 1 to 2 years. Now, there's nothing in particular wrong with this analysis. It's very common to integrate different methods. Nevertheless, this integration is informal. The genetic side of the analysis does not talk to the niche side of the analysis and vice versa. So how can we formally integrate these methods to infer species biogeographic histories? We're going to show you a way forward by focusing on green ash, a very common tree in eastern North America. Our primary questions are, where were the locations of refugia during the last glacial maximum, and at what rate did the species migrate as glaciers receded? First up is inference using pollen. Pollen data comes from cores of lake beds, and as such it usually reflects what was growing around the lake at the time. So what we need is a model that will interpolate spatially and temporally into regions where we don't have samples. I encourage you to check out Alyssa Brown's talk on this pollen model in the same organized oral session. The model is incorporated in the PGR package for R by her, John Tipton, and Andrea Dawson. Here are the predictions of a simplified version of the pollen model. As you can see, it suggests that the epicenter of ash abundance around 16,000 years ago was somewhere around the Ohio River Valley. The predictions out in the ocean are due to the fact that there actually was land during that time, but it's since disappeared due to sea level rise. What this suggests is that there was one very large periglacial refugium and perhaps a much smaller one out in the east coast. In the second line of analysis, we applied ecological niche models. We used two different algorithms, two different global circulation models for past paleoclimate, and two different background extents. We also corrected for undersampling of certain areas. This gave us 8 different possible outcomes. But I'm going to focus on the two models that were the most different in the past. The one on the left here is created by Maxent. It depends how you interpret it, but you could say, for example, that it represents one very large refugium. On the other hand, perhaps a species was relegated to a very localized spot of very high suitability in the southwest. The map on the right was created using a generalized linear model. Again, you could interpret this as suggesting that there was one very large refuge. However, it's also possible that it suggests that the species was relegated to a periglacial refuge in the west and a southeastern refuge. The take-home here is that even within a method, there can be notable disagreements. It's easy to see that the pollen model and the niche models are inherently spatial. This is how we go about spatializing an estimate of the genetic trajectory of the species. Here I have a cartoon of various populations sampled across a landscape. We're going to presume we have a blue genotype and an orange genotype, and we're going to hypothesize that the species hunkered down during the last glacial maximum in a western refuge and an eastern refuge. Now, as you might imagine, there are an infinite number of ways for the species to spread out from these two refugia and recreate this present-day pattern of genetic diversity. For example, we might posit that the western refuge, which today is primarily occupied by the blue genotype, was in the past primarily occupied by the orange genotype, and vice versa. This is much less likely, though, than a scenario that begins with orange in the east and blue in the west, and that's fairly easy to intuit. What's more complicated, though, is intuiting the relative likelihood of all the intermediate stages. For example, we might posit that the species began to expand from the western blue refuge and then later from the orange eastern refuge. But how do you assign a likelihood to this compared to all the other trajectories that you could think of? To do that, we employ approximate Bayesian computation, and this is how that proceeds. First, we begin with a cell-based demographic simulation where the species starts out in a refugium with population n sub b, or effective population size. The species migrates across the landscape either through short distance movements to neighboring cells or through long distance movements to cells that are farther away. Writ large across eastern North America, this is what that looks like. In this particular case, we posited that the species had 3 different refugia, one in the southwest, one in the southeast, and one that was periglacial up north. The color coding here indicates the first time at which that cell was colonized as the species spread out from those refugia. And now, knowing which population begat which population, we overlay a backward time coalescence simulation on the history of the population colonizations in order to spatialize the genetic component. So the two different kinds of simulation, the forward time demographic simulation and the backward time coalescence simulation, are inherently stochastic. They're also governed by a range of free parameters. You can see them here. Basically, we allow the computer to pick those values for each simulation across a certain range. However, for some simulations, we choose to start with a refuge located either in a northern periglacial area, a southwestern area, or a southeastern area. For this particular analysis, we did not look at the two-way combinations of refugia. We ran 10,000 simulations for each refugium scenario. Given that each of the simulations is inherently different from any of the others, how do we select those that are more likely? So if you recall my cartoon using the blue and the orange genotypes, the idea is to see which scenarios and which simulations best recapitulate the observed pattern of genetic diversity. So for green ash, we have 21 different populations, each scored for 1,000 SNPs. And I want to acknowledge the contribution of Jean Romero-Severson and Andy Noakes, who actually did the fieldwork in DNA extraction. So from the DNA data, we calculate about 2,000 summary statistics as targets for the simulations. These summary statistics relate to local genetic diversity, interpopulation genetic diversity, and spatial patterns of genetic diversity. We then take our tens of thousands of simulations and see which ones most closely replicate these summary statistics. Here's an example on the left. This is just in 2-dimensional space, but of course we're looking at 2,000-dimensional space. The idea here, of course, is that the simulations that most closely replicate reality capture some element of the truth. This whole method is known as approximate Bayesian computation. Approximate because we can't write out in closed form an actual likelihood. We use simulations instead. Using this technique, we can then estimate posteriors for some of those free parameters. For example, a very common question is how important were long-distance dispersal events to the movement of species? So here in the upper left-hand corner, we're plotting the prior and the posterior for the ratio of long to short distance events among the accepted simulations. And as you can see, somewhere between every 2 and 4 in 1,000 colonization events arises from long distance dispersal. Now looking at the plot in the upper right, you can see that we can estimate the carrying capacity or effective population size of the species in a cell. This isn't to say that approximate Bayesian computation can answer everything. For example, we do a fairly poor job of estimating the effective population size prior to the last glacial maximum. So now let's see what ABC says about the location of refugia. Of the top 10% of the models that best matched our data, only about 7% began with a refuge in the southwest and another 7% began with a refuge in the southeast. 31% of those simulations began with a paraglacial refuge in the north. However, 56% of the simulations began with all refugia together. Now, it's not that we're asserting that the actual refugia were in these locations. A more robust interpretation is that a single refugia model is not very well supported, and at least one of those refugia was paraglacial. Now, these simulations are so far fairly generic in approach. For example, we assume that if a cell is not occupied by ice or water, it's fully colonizable. Likewise, other than melting the glaciers, we assume that climate has no role. We can also use cross-validation to examine how well we can identify the different scenarios. Here we take the summary statistics for the 10,000 simulations we ran for each scenario. Then we use a random forest algorithm to see how well we can differentiate between the different scenarios based only on their summary statistics. So what I'm showing you here is the proportion of each of those 10,000 simulations for a particular arrangement of refugia that could be identified as belonging to that arrangement based solely on their summary statistics. And as you can see, the values along the diagonal are fairly high, especially for the paraglacial refugium and for the case when we have all three refugia at the same time. This gives us confidence that our summary statistics are capable of enabling us to differentiate between scenarios. So in summary, what we found was that while each of the methods generally agreed in terms of the number of refugia, they are very different in terms of the location of the refugia and their nature, whether they were more in the south or more paraglacial. Obviously, there's a lot we could improve on, but the take-home message here is that an informal integration, that is comparing them by eye, doesn't really work. We need a formal integration across statistical methods. So it should be evident that informal integration of these different types of data and the respective methodologies is, in the end, informal. There's no crosstalk between the different methods. And even though in general results from different methods might seem coherent, the specifics can be dramatically different. So our main conclusion is that the field is ripe for a formal integration of these different methods. One way to do that would be to merge, for example, the niche models and the genetic simulations, or the pollen vegetation models and the genetic simulations, using the former as constraints on carrying capacity for the latter. And it's our firm belief that only a full accounting of all sources of information and uncertainty will allow us to really infer the biogeographic history of species. Thank you.